Good afternoon and welcome to Biobrain's National Science Week webinar on antibodies, vaccine and the COVID-19 pandemic. My name's Caroline Cotton and I'm very excited to be hosting you tonight. I'm welcoming people from all over Australia, students, teachers, researchers, doctors and other professionals. The coronavirus is something that's impacted us all this year in ways that we could have never imagined. From not being able to see family and friends, not being able to go to work, students learning from home, and teachers teaching them from home, or exercise being limited to no longer an hour currently in Victoria. However, on the flip side of this, it's enabled us to have this webinar today with people tuned in from all around Australia, so welcome. I'm very excited to have two scientists here today who are working very closely on the SARS-CoV-2 virus. Before I introduce our two guest speakers, I'd just like to run through a few general housekeeping things as we've got over 150 people registered. So the session will run with hearing from both presenters, 15 minutes each, and then after that, I'm sure you'll have lots of questions. So if you could please put your questions in the chat and we'll have 15 or 20 minutes at the end to go through the questions. Please leave the chat box empty apart from the questions. And if you could all make sure your microphones are on mute throughout the session so we can avoid any background noise. Our first speaker will be Dr. Simon Preston. Simon's a Senior Postdoctoral Research Fellow at Walter and Eliza Hall Medical Institute. He completed his PhD in microbiology and immunology at the University of Melbourne, focusing on host pathogen dynamics, and has been working on translating his findings into therapeutics ever since. He has worked on discovering new treatments for HIV, HBV, TB, and most recently SARS-CoV-2. His passion is to translate basic discoveries and new and innovative therapies to treat infectious diseases. Welcome, Simon. Our second speaker will be Dr. Brad McColl. Brad works at Affinity BIO, a company that is developing antibodies-based therapies to attack targets located inside cells, such as mutated proteins that cause cancer or viral proteins. Recently, Affinity BIO has also been generating antibodies that bind to the spike protein of the SARS-CoV-2 virus and prevent it from entering human cells. I now welcome and hand over to Simon for his part of the presentation.
Can you hear me, Caroline?
Yes, thank you.
All right. Well, thanks for the introduction, Caroline, and thanks for inviting me to speak today. Caroline was actually my biology tutor in BCE, so a few years have gone since then. So I'll just start by introducing the coronavirus a little bit and speaking about how this virus causes the disease we've come to know so well. So COVID is the disease that's caused by SARS-CoV-2, also known as the novel coronavirus. The SARS part stands for severe acute respiratory syndrome, because this is often what happens when we develop severe disease, and those are the people that are the ones that have to go to hospital with this virus. And looking at the virus itself is a really nice illustration of the basic components of the coronavirus. We see in the middle here, it's genetic material, also known as RNA, and that's really the blueprint for how the virus copies itself. We also have nucleocapsid and envelope proteins, which form the outside shape. And very importantly, we have membrane proteins that sit on the outside of the virus. And that's the part of the virus that's exposed to the immune system, and generally the parts of the virus to which our immune system generates antibodies to once it sees this virus. And the spike protein, which Caroline already mentioned, is the one in green there that sits on the surface. And this spike protein is actually what gives the virus its name, coronavirus, corona meaning crown. Now, I've taken this illustration from the Washington Post because I really do like this one. And it's just a way of showing you what really happens in the viral life cycle once it infects a human. So all viruses must infect host cells in order to replicate their genetic material and make copies of itself. So here I've shown when the coronavirus goes into our respiratory tract, it actually interacts with our respiratory cells through its spike protein, and it binds to what's called the ACE2 receptor on the surface of cells within our respiratory tract. That causes the cell to take up the virus, which leads to a release of its genetic material, its RNA. And the really clever thing the viruses do is that they hijack the protein building machinery of our cells to make copies of the virus, make millions and billions of copies of the virus, which are released from the cell and go on to infect all other cells within our respiratory tract. And it's that real repeated infection and reproduction that leads to the production of virus that can then be spread from one person to the next. So here I'm just showing the course of infection. So the very start of the graph here is when that initial infection occurs, when someone gets exposed to the virus. And through that round of infection, we see the viral load go up in the blue line. And we start to develop symptoms, if at all, around day 5. And I say at all because sometimes people don't even realize they have symptoms, as you may have read, or even someone you know. And that's why we call it, it's relatively silent, but it can also be deadly. And part of that is because this virus is highly infectious. It's actually more infectious than the common cold or flu, and that's why it just is transmitted so well. But the other reason it transmits so well is because whilst we may get symptoms around day 5, it's actually days before that we become infectious. So you can spread virus to multiple people, multiple close contacts within your family or at work, even before you know that you have it. And that's what's really led to this virus really taking over the world the way it has this year. So it's not until about day 6 or 7 when our immune system starts to kick in, our adaptive immune system. And I've shown here some immune cells, and as they start to increase in number, some people will develop severe disease. And it's not actually the virus that causes this severe disease. It's actually the release of proteins called cytokines from our immune cells that causes these symptoms. But once this occurs by 1 to 2 weeks after infection, and regardless of whether you get mild infection or severe infection, our body starts to produce a degree of immunity to the virus. And it's this interaction of B cells and T cells that lead to the production of antibodies, which we believe might be protective to reinfection. They generally are for other infections, but it's so early on in this pandemic that we don't truly know whether or not we'll be protected after we're infected. It's likely, but that's still to be borne out. So what I'd like to speak to you about today is the research that us and those we collaborate with at WEHI, which is an acronym for Walter and Eliza Hall Institute. And I'll be speaking to today on our efforts towards diagnostics, drug discovery, clinical trials, as well as some disease susceptibility and biologics. So the first thing I'm speaking about is diagnostics, in particular a diagnostic device that we're developing. So one of the first things we noticed is that Australia was too slow to get testing up and running. We could see that it would have been really useful if at airports or ports of entry where those cruise ships arrived at Australia, if we'd had something there that could screen passengers before they arrived, we may have prevented a lot of the seeding of the virus that we had that's led to where we are now. And so what we believe is that we need to develop an Australian-made cutting-edge diagnostic platform that has the flexibility to rapidly adapt and recognise novel infections. Because it's likely that this won't be the last novel infection, and it's likely this won't be the last pandemic we have. And what we're developing is a point-of-care device which we believe can deliver a result in 15 minutes. So right there at the moment, rather than if any of you have been swabbed for the coronavirus, I know I've already been swabbed 4 times this year, it can take a couple of days to get a result back. And in some other countries, it can take a week or 2. So getting a result straight away means that we won't be going and interacting with other people. We won't be spreading the virus. And it's a really important control measure for these kind of pandemics. And we believe that we can develop this device that can be implemented at airports or schools, for example, And it's based on this technology called CRISPR. Very briefly, all that means is that we have a guide RNA, which is fused to an enzyme called a Cas enzyme, and that guide RNA is highly specific for the coronavirus, the RNA target. And if there's virus in that swab that gets taken and put into this machine, it'll recognize it. That Cas enzyme activates, and it cuts in between these two things called a quencher and a fluorophore, and it releases light. So basically, it's just a device that can recognize the virus, releases the light signal, and you've got a result in 15 minutes. We're also working with the National Drug Discovery Centre, which is located at Weehai. And what we're doing here is that, and no person is doing, we're actually getting this amazing robot to do this. And what it's doing, it's screening thousands and thousands and thousands of potential drugs or compounds that might have activity against coronavirus proteins. That could be the starting place for development of a new antiviral, both for coronavirus and for future viruses. And what we're doing, we have a high containment PC3 facility at our institute, and we're testing some of these compounds, as well as those that have already been approved for other infections or other conditions, to see if they have activity against coronavirus. And these are two of my colleagues here, James and Cody, who are doing a lot of the hands-on research for us. We're also developing a new testing platform to screen some of these drug compounds. And these are three-dimensional mini-human lungs in a dish, which we also call organoids. And we think that this is a really great model to screen new compounds which might have antiviral activity. Another thing we're working on is that we're conducting a gold standard clinical trial to test whether a drug called hydroxychloroquine can prevent COVID-19. We're conducting this trial in healthcare workers, frontline healthcare workers. You might have heard of this drug before in the media. It's had a lot of attention, but I'll tell you a bit more about it. It's most commonly used to treat an autoimmune disease called lupus, and less often it's also used to treat malaria. For the trial, we're recruiting 2,250 healthcare workers, and these participants will be randomly assigned placebo or active drug. We're currently recruiting participants at the moment, and what we're looking for is we're looking at whether or not they contract COVID-19, which is happening. You would have heard that there's a lot of healthcare workers. There's over 1,000 healthcare workers in Victoria alone that have contracted the virus, and our goal is to really try and see if we can help to find something that can prevent that. What's important to point out about this study is that no other study has conclusively addressed whether pre-exposure prophylaxis, such as this, it's also known as PrEP, works. Only treatments has been addressed, which we know now doesn't work. But the rationale for prevention is similar to what we've seen with HIV treatment. We know that PrEP prevents infection of HIV, but the very same drug or drugs have no effect on the virus once the person's already infected. So we're looking at prevention, and that's ongoing. And we're also testing human antibodies, as well as these alpaca nanobodies, which they generate, these tiny fragments of antibodies, which are a really interesting new area for biologicals. And we're using these in treatment and prevention of COVID. We're screening human antibodies that have been generated at Weehai, which recognize SARS-CoV-2. We are also screening nanobodies that recognize the virus, and we're looking at seeing if these can prevent infection of cells in a dish, prevent infection in animal models of COVID-19 in our PC3 facility, as well as to see if these antibodies or nanobodies can treat existing infections with the virus. So at this point, I'll hand over to Brad to describe how our biologicals are being generated for COVID-19, as this is his expert area.
Thanks very much, Simon. I don't know if you can call me an expert, but I'll do my best. So yeah, I work for Affinity BIO. We're a small antibody company in Melbourne. We have mainly a focus on oncology, on cancer-related antibodies, but given the worldwide nature of COVID-19, earlier this year we kicked off some screens to chase antibodies against the COVID-19 virus, and I'll talk a little bit more in depth about those. But first, just a brief introduction as to the nature of antibodies. So antibodies essentially are proteins made by the immune system. There are various kinds. The common one that you see in all the textbooks is this Y-shaped protein you see on the screen in front of you here. So that's known as immunoglobulin G, or IgG. This is one of the most common antibodies you'll find in the body, this class. It's made of 4 protein chains, 2 heavy chains and 2 light chains. And those proteins are divided up into structural domains, which are referred to either as constant or variable. Now, the variable domain defines the target of each antibody. There's regions of protein sequence in those domains which differ between all the antibodies, and it's that structure which defines the binding to the target protein. Now, the function, what an antibody does in the immune system, I've tried to diagram out here on the right-hand side of the slide. So immune cells secrete antibodies in the blood where they float free throughout the body and free to bind any foreign proteins, pathogens, germs, viruses that they might encounter. And the result of that binding, there's a few effects. One of them is simply to interrupt the functioning of whatever that pathogen might be, because suddenly it's got an enormous antibody stuck to the surface of it. And also the constant regions of the antibody act as a flag for the immune system. And cells known as macrophages can recognize pathogens that are bound by antibodies, and it marks them for destruction by those macrophages. The next slide, please. So just to talk a little bit about the biology of antibodies and how the immune system makes them. Antibodies are produced by this group of cells you can see circled here, the B cells, which Simon mentioned earlier. B cells are made by the stem cells found in the bone marrow, which generate all these other cell types as well, other types of immune cells, red blood cells, which carry oxygen around the body. But it's the B cells that we're really interested in right now. So the bone marrow generates about 20 million B cells every day, and significantly, every B cell produces a randomly different antibody. So during development of each B cell, the genes for the antibodies inside the B cell undergo a random shuffling and mutation. And so those variable domains of the antibodies come about as a result of this random process. And so the body's constantly producing, these huge numbers of random antibodies, which are in the first stage, they're they're displayed on the surface of the B cells, and those B cells are then free to diffuse out into the blood. And now, look here on the process I've outlined on the right-hand side of the slide. This random collection of B cells with their antibodies on the surface, when they encounter an antigen, which is might be a protein from a bacteria or a virus, when they encounter a protein that binds to that antibody, that B cell then undergoes a process of activation where it replicates itself many, many times, and those daughter cells are then differentiate into one of 2 cell types, either what are known as plasma B cells, which then secrete many, many copies of that antibody into the blood, where it can go out and attack that particular pathogen. Or also a different subclass called memory B cells. And these are very important in terms of the functioning of vaccines. Those memory B cells are very long-lived cells in the body. They keep that antibody specificity. So in the event that the body encounters that same pathogen again, that group of memory B cells kicks into action very rapidly and generates a large population of plasma B cells to give a rapid antibody response to that pathogen that we've seen before. And this is what happens when someone's given a vaccine. Assuming the vaccine works well, they develop a population of those memory B cells. And so later on, when they encounter the genuine virus or bacteria that they've been vaccinated against, they've already got that antibody response primed and ready. So next slide, please. So I just wanted to touch briefly on the number of vaccines that are currently in development around the world. This is a recent document from the World Health Organization summarizing the state of play in vaccine development for COVID-19. As of last week, they listed 29 vaccine candidates that had entered into clinical development, either phase 1, 2, or 3. That means they're basically being tested in human volunteers rather than just being tested in the laboratory. You'll see the very first one on the list is the AstraZeneca Oxford vaccine that's being touted by the Australian government in recent days. There's also about a further, it's somewhere between 100 and 150 other vaccines that are in earlier stage development but haven't yet entered clinical trials. So hopefully, within the next 6 months, we're going to start seeing a rollout of vaccines around the world. But just to come to the question of what we at Affinity BIO do, we can ask, is there anything else we can do aside from vaccination? Let's go to the next slide, please, Caroline. So as I mentioned, we're an antibody company, so we're interested in antibody therapy. And you might have heard in the news people talking about using convalescent plasma to treat patients. So in that case, you're taking blood serum, which is the fraction of the blood which contains the antibodies. You're taking that from a recently recovered patient who still have a high concentration of antibodies against coronavirus in their blood. And then that plasma fraction is administered to patients who are still actively who still have an active infection, with the idea being that you're adding a large dose of antibodies against the virus into this sick person with the aim to assist their immune system to eliminate the infection. There are a number of issues around this to do with you have to have a supply of recently recovered patients. They only maintain their antibodies for so long. Quickly after you recover from an infection, the antibody concentration in your blood drops back down to normal levels. There's also issues of safety once you start talking about transferring blood products between one person and another. This is a non-standard approach also because not everybody makes the same antibodies, so you can't make an accurate estimation of what the effective dose might be. There's a lot of issues around this, and it was interesting to see in the media today the FDA in America have actually put a hold on their process of approving this particular type of therapy for the time being, given there's actually not a lot of data to show that it's really an effective therapy. So can we use an antibody generated in a laboratory where we can have a known supply of antibodies which behave in a well understood fashion? We can learn about what the appropriate dosage might be. We can have a highly purified, available supply, and we don't have all those potential safety issues involved with blood transfusion. Next slide, please, Caroline. So this is coming to what we do at Affinity BIO. And how you generate an antibody in the laboratory. It's a similar approach to how the immune system does it, in that you generate a very large number of random antibodies, and then you go looking for the ones you want. In order to do that, we use a different kind of virus called a bacteriophage, which is a type of virus that only infects bacteria. These are very well understood. People have been studying bacteriophage in molecular biology for about 60 years now. And so there are many tools and tricks that we can do with these bacteriophage. One of the things we can do is insert the gene for a foreign protein into the bacteriophage DNA, and then the bacteriophage will produce that protein and display it on its on the surface coat of the bacteriophage. So the phage DNA is enclosed in a protein capsule, and then we can make it display this new protein on that protein capsule around the phage DNA. And when by doing that with a gene encoding an antibody, you then get these little bacteriophage decorated with antibodies. And so we've generated a library in bacteriophage doing just that. So we have the current library that we work with has about 100 billion different antibodies in it. And the significant point is these bacteriophage don't just contain the antibody, but they contain the gene encoding the antibody. So you don't just get the antibody out of the library, you get the instruction set for making more of it as well. That's the really important point. And so when we want to find find an antibody in our library which recognizes the protein that we're interested in this case, the coronavirus spike protein, it's kind of like going fishing, where we just essentially dangle the bait protein into the library and we pull out the bacteriophage that stick to it. And we repeat this process a few times to get a complete screening of the library and to make sure we get the optimum antibodies that bind to our target protein. And then we can isolate the genes encoding those antibodies and work further with our candidates that way. So, move the next slide, please, Caroline. So yes, we kicked off a screen in, I think it was late March this year, looking for antibodies that would bind to the coronavirus spike protein. And we didn't just want antibodies that bound to the spike protein, we wanted antibodies that would inhibit that interaction of the spike protein with the ACE2 receptor that Simon mentioned earlier. So that's the surface protein that the coronavirus binds to as the first stage of infecting cells. So we wanted an antibody that would interrupt that process. And so we pulled out about 100 different good antibodies out of our library screen, and then we tested we eliminated those ones which didn't affect the binding of the spike protein to the ACE2, and then we took we came down to 3 best candidates, and we tested those in an assay to determine which of those was most effective at blocking the binding to ACE2. And without going into the details of how this plot beside you, essentially it's just measuring the binding of the virus spike protein to its ACE2 surface partner. And the top, you see the negative, that's just the background noise in the assay in the absence of any spike protein. And then the next bar in black shows the maximum binding that we observed when we didn't add any antibodies into the assays. And then we see that we've tested our 3 best candidates at different concentrations. You can see as we increase the concentration of each antibody relative to the spike protein, we're getting a reduced binding of the spike protein to the ACE2 surface receptor. So that was really encouraging. But it's just that was just a test tube experiment to look at our antibodies. We then we sent those to the Doherty Institute at Melbourne University, who tested those antibodies against genuine coronavirus in an infection assay on cells in a petri dish, and showed that we could get very good inhibition of the virus infection. And there's further testing those antibodies are undergoing further testing at the La Jolla Institute for Immunology in San Diego. We're expecting further information about those in the next few days. The idea really would be that these could be used as a stopgap to treat people who have either, who have been infected with coronavirus, or also people who might be at high risk of being infected. Like in the case, we've seen a lot of infections in aged care facilities, and when you get an outbreak like that, you could potentially go in and dose people with these antibodies, and they'd be protected or be at reduced risk of being infected for 6 to 8 weeks with each dose of antibodies. So that's where we're standing at Affinity BIO at the moment. Caroline, can I hand back over to you?
Thank you very much, Brad. Thank you very much, Simon. Has anyone got any questions? If you could put them in the chat, please. I can't see any there. Oh, here's a question. from Komputukura, sorry. Hi, Simon. What animal model of SARS-CoV-2 infection will you use to investigate the nanobodies?
Yeah, so thanks for the question. So we mainly use a mouse model, and we've got a couple of different options to choose from, but basically we've got to do a trick where we can express the human ACE2 receptor, because the virus won't actually infect mice. It's very human tropic. So we have mice where they express the human ACE2 receptor, and then they do become permissive to the virus. And so what we can do is we can either give those nanobodies or antibodies before infection, like Brad was just saying, like could be used in a nursing home to see if we can prevent infection. But likewise, we could also infect the animals first and then add the antibodies later to see if we can ameliorate disease that way.
Thanks, Simon. Has someone got another question they want to put up? I've got one for you, Simon. How long do you expect it to take before you think you'll have some results from the COVID Shield trial?
So at the moment, we've been recruiting for probably about 2 months now, and it sort of depends on how many positive cases we get within the cohort. We have a number that we're looking to get that will require for statistical power to calculate whether or not there's the effect size that we want that could prove whether or not the drug is working. So we haven't got to that number yet. But what we'll be able to do is when we get there, we can do an interim analysis. So we don't have to wait for all participants to be enrolled. We just know that we can do an interim analysis. And at that point, which we're not quite there yet, but hopefully in the next few months, we'll be able to give you an answer.
Great. Thank you very much. Someone else got a question? Any of the students out there got any questions? Or anyone else in the audience? Oh, here's one from Kim. With the COVID Shield trial, how do you correct for the differences in infection control procedures with different institutions, which could influence the infectivity rates?
That's a really good question. We don't see it as necessarily when we have so many people in the trial, we sort of and across different hospitals, we don't assume, but there's going to be a similar level across all of the Australian hospitals, and there's going to be times when people are exposed or not, and we take that into account with our statistics that we use to determine how many positive cases and in which arm will be enough to determine or validate the hypothesis. So that's all taken into account for the statistics of the trial.
Great, thank you, Simon. There's a question from Susan. This is more to me. Yes, Susan, we will be making the PowerPoint presentation available to you. So if you contact me at the end of the presentation, send me an email, I can forward you a copy of the slides so you can show your students. Has someone got any more questions? I guess one question I'm sure people want to know is, Brad, perhaps you can answer this one. You sort of touched on it briefly, but what makes the SARS-CoV-2 virus so virulent and deadly compared to the influenza virus that we've seen for years and years?
Yeah, it's an interesting question. Really, if a great way to consider that is to think about the SARS epidemic that we had in the early 2000s, which is from a very closely related coronavirus. And it even has the same cell surface receptor as the COVID-19 virus. But if you compare what we call the affinity of that interaction, I'm not talking about the company here, I'm talking about the scientific concept. The affinity of the COVID-19 spike protein for the ACE2 receptor is very much higher than the original SARS coronavirus spike protein. And so the consequence of that is that the SARS virus can bind to its surface target, and it increases the chances that you'll get infected from a given dose of virus. The earlier SARS virus, you really had to have a very large dose of the virus in order to be infected, whereas it seems like the current COVID-19 virus, it doesn't require nearly such a high dose in order to infect someone. So I think that's a very there's probably other factors at play, but I think that's a very important factor in determining how infective the virus is.
I could have a go at answering that one too, Caroline, if that's okay.
Please.
Yeah. I was say I think this is kind of like the perfect virus. When you look at SARS-1, it was actually caused pretty severe disease in everyone that got it. And so when you did become very sick, you were in bed and you couldn't interact with other people. So it actually didn't transmit to as many people. But for the reasons that Brad was just describing in terms of its infectivity, for whatever reason, the symptoms of COVID aren't as severe. And so someone who only has a bit of a sniffly nose or a tickly throat and passes it off as nothing might still go to work and transmit it to 5, 10, 15 people. And that's been the really clever thing, for lack of a better word, for this virus, is that it's really infectious. It doesn't cause severe disease, but just enough and in a fraction of people. But when you infect so many people, that fraction ends up being hundreds of thousands of people. And it's just the combination of those factors which are just peculiar to this virus. Are you on mute, Caroline?
Sorry, it's just with the investigation into antibodies binding to spikes, what is the likelihood of COVID-19 mutating to make this investigation redundant? How do you assess for this?
Is that to me, I guess?
Yeah, well, I think so.
Yeah, but look, it's a good question, and whenever you come up with a new medicine, no matter whether it's an antibiotic or an antibody, you're always chasing down that Darwinian evolutionary rabbit hole. So that's not a problem that's unique to this particular situation. We pulled out our 3 best antibodies, with our thinking being that you wouldn't administer them just one at a time. You would administer them to a patient as a cocktail, a mixture of multiple antibodies, in order to minimise the possibility that the virus is going to escape by having a single mutation. If you put the virus in a situation where it's got to mutate away from several antibodies, you're much less likely to see those sorts of escape mutants arise. So that, and I should point out, we're not the only company trying to do this. There are other companies as well, and they're taking that sort of cocktail approach as well.
Great, thank you, Brad. Next question. Binding to the surface proteins and preventing access to our body cells, is the virus killed or simply unable to penetrate and infect immune cells, and could they potentially unbind?
Yeah, we actually, in our screens, we selected for antibodies that have a very long, what's called a very long off rate. So they bind to the spike protein and then they stay there. A lot of antibodies will bind and then fairly rapidly fall off their target. We've deliberately selected for antibodies that were very stable, and we've tested them for several hours and seen that they remain attached to the spike protein. And then the infectivity assay that we did at the Doherty Institute, the virus was put on the cells for 5 days with our antibodies, and we could still see at the end of 5 days that the cells were remaining uninfected. So yeah, we've given that some thought, and hopefully the antibodies that we pulled out are sufficiently stable that that's not going to be that's not going to be a problem.
Great, thank you. Our next question is from Jay. Our Prime Minister has recently signed a letter of intent to acquire the vaccine for COVID-19 from University of Oxford. Are you currently in collaboration with them on the release of this vaccine? I guess that's to either of you.
No, we're not. That's nothing to do with us.
No, neither with us.
Okay, thank you. Next question for Brad from Comptictor. Sorry about your name pronunciation. Do you think that if these synthetic antibodies are widely overused, would the body start to mount an immune response and develop antibodies against the synthetic antibodies, rendering them useless eventually?
Yeah, it's a good question. Our particular antibody library is what's called fully human. So the scaffold of the protein, those constant domains, and the constant region of the variable domains are all human derived. So those are sequences that shouldn't provoke an immune response because the immune system's already seen them and recognizes them as self. So we've taken steps to avoid that when we set up the library.
Great, thank you. Here's a question from Pam. Do all cells have an ACE2 receptor or just cells in the respiratory system? Very good question.
I can try and answer that one if you like, Brad.
Yeah.
So a lot of, well, pretty much all cells in the body do express the ACE2 receptor to a certain degree. We know that within the respiratory tract, that there's actually a higher level of ACE2 expression higher up in the respiratory tract, and it decreases as you go down, and that could be part of the reason why, with that affinity and that high expression in that start in our nasal passages, that it really is easy to infect in that infectivity level. But it is expressed around the body as well, and that's why you might have read that there are you can find the virus in the blood, in the heart, in the kidneys, and all these other complications that we're starting to hear about. And that's probably got to do with the fact that the virus can get into our bloodstream as well and infect other cells within the body with basically new complications that are coming out every couple of weeks.
Great, thank you, Simon. Here's another one for you, Simon, from Kim. How close are we to having a CRISPR technology available for rapid testing in Australia?
Well, we can do it right now in the lab, but being able to put it into a device, get it through regulatory approval might take a little bit longer. We're aiming for possibly the end of next year to have something that's able to be used for regulatory purposes, but unsure when it might get to market. But I don't think even by next year, COVID will completely have disappeared. We may have it under control, but it's likely that we'll get spikes here and there, and will be quite important over the coming years to be able to test people quickly at ports of entry, I think, for many years to come.
Great, thank you. Next question from Gina. Does the mutation rate of COVID-19 change the effectiveness of the possible vaccination? Is it possible to continue mutating at a rate that we can't efficiently find a vaccine for the newer forms? Brad?
Yeah, good question. It's a little bit outside my area of expertise. Generally, though, with a vaccine, you're targeting multiple points on the virus simultaneously. So even if the virus mutates in a way that eliminates one aspect of the vaccine, there's generally other sites that the vaccine is delivering and that the immune system can recognize. So I think it's unlikely that it would happen in a very rapid, very short period of time.
Yeah, so coronaviruses have a much lower mutational rate than HIV, for example. So HIV generates a lot of mutations very often, and that's why they initially found it very difficult to get any drugs that would work, and that's why they needed to have triple antiviral therapy initially for HIV for it to work. Coronavirus is very slow in that respect. So there has been one or two mutations that have become dominant, but you'll see them that they start to become dominant and they take over. Like all in Australia at the moment, it's mainly one mutation. And the fact that there's only one tells you that there's not a lot going on. And if there are, that they're not very dominant. And so I think it's unlikely that we'll have an issue with mutations happening that will prevent a vaccine from working. And as Brad said, a lot of the vaccines that are being produced don't just have one protein and one antibody that it will generate. There'll be multiple peptides that'll be recognized. And so even if there's a mutation in one, it'll probably be captured by the other.
Great, thank you. Next question from Endem. Do you think the fast tracking of vaccine trials will have an impact of the vaccine quality, seeing as they normally take a few years to develop?
Yeah, well, I'd hope they're being held to the same regulatory standards as a traditionally developed vaccine. So I would hope not. There's also there's a number of other technologies being used in developing the vaccines, and I'm thinking about a company called Moderna that's delivering viral RNA rather than protein. But and using those new technologies can speed the development process. Yeah, it's a good question. I would anticipate that the regulatory authorities are watching this very closely to ensure that that's not going to be the case. Simon?
Yeah. I'm sure that they'll all be held up to the required standards. I think what we're seeing now is, for the first time, every major pharmaceutical company and almost every lab is working on coronavirus, and we've never seen such a concerted effort to tackle the one virus or the one pathogen. And so I think they're all being done properly, and I think this will be the best chance to get a vaccine quickly. I mean, even going back, I was looking today that I think it was the mumps vaccine was made in 4 years back in the 60s. But you've got to think that we're in 2020 now, and the technology is so far advanced than what it even was 20 years ago. So our capacity to generate new vaccines is also much faster. So sometimes comparing it to historical sort of 10-year timeframes isn't exactly equivalent. So I think they're doing it the right way, and I'm sure there'll be a few of them that work.
Great, thank you. I think I missed a question from Eliza about binding to surface proteins and preventing access to our body cells. Is the virus killed or simply unable to penetrate and affect immune cells? And could they potentially unbind?
Well, yeah, I sort of touched on that, but then once the virus is coated with antibodies, it's then really exposed to being digested and cleaned up by immune cells. If it's sufficiently coated with antibodies that it's prevented from infecting a cell, then it's really just debris as far as the body's concerned, and the regular immune surveillance should clean that up.
Yeah, great, thanks. I think you might have almost answered this one. The next one from Yoga. Can ACE2 receptor blockers be used to block the attachment of SARS to the cells expressing the receptors?
Yeah, that's an interesting one. There are people looking at making antibodies against ACE2, and so sort of hiding the receptor from the virus. I'm a little bit skeptical about that approach because, and Simon, you might be able to fill me in on this. ACE2, I think, that's involved in blood pressure regulation. Is that right?
Yeah, angiotensin converting enzyme is what it stands for.
Yeah, so messing around with what is perhaps an important component of maintaining your blood pressure might not be the most straightforward approach. There are groups who are working on using fragments of ACE2 that could be injected into the body, which float around and which act kind of as decoy receptors for the virus. That's another approach that people are looking at, but I haven't heard a great deal about how that's progressing.
Okay, great. Thank you. Here's another question from Julia. After a swab test is taken, what is the current lab process of detecting COVID?
I guess I can answer that one. So I've also, like I said before, I've been swabbed, I think 4 times now myself. So yeah, but when they do that, they take the swab and then they take it to the lab, and then what that swab actually gets put into some liquid, which we call media. And what they do is they'll take that liquid out of the tube, which would have virus on it if you were positive. And then they put it into a plate and they lyse the cell. And then what they do is they use primers, which can bind to the virus. And then they undergo a process called polymerase chain reaction, or PCR, where it goes through this heating and cooling process that will amplify the virus if it's there and will emit a signal. The difference between what I was describing before with CRISPR, even though they both use primers, the CRISPR can happen in 10 to 15 minutes, whereas a PCR takes 45 minutes to an hour, and it also has to be taken off to a lab. But yeah, so they lyse the virus, they amplify the genetic material, and then it's detected in a machine.
Thanks, Simon. I've got another question that's come in. Is the WEHI vaccine designed to prevent infection or inhibiting the growth of the disease?
So at WEHI, we're not particularly working on a vaccine per se, but we are looking at several different avenues. So the clinical trial is looking at trying to prevent infection. And we're also screening the antibodies, similar to what Brad's doing. So we're basically having a multi-pronged attack. So we're not sort of putting all of our eggs in one basket. We're trying to look at different aspects of it and see which one might be the most successful.
Great, thank you. Has anyone else got any more questions? Just got a couple more minutes. We could have 1 or 2 questions if someone had any. Well, if we don't have any more questions, I just want to thank everybody very much for attending and participating in Biobrain's National Science Week webinar. And particular thanks to Brad McColl from Affinity BIO and Simon Preston from WEHI for the insightful presentations and answers to the questions. So hopefully everybody's learned something out there, some more than others. Some people are obviously working in the area, some people are students studying it for their biology, obviously teachers. So thank you very much for everyone for your participation, and particular thanks to Simon and Brad, and have a great Science Week. Thanks so much.
Thank you.
Bye.
And if you'd like a copy of the presentation, you can email me. Thank you.